Welcome to PAPERI Bright Minds Series, where we spotlight the pioneers shaping the future of genomics and genetics. This session features Dr. Marina DiStefano, a board-certified clinical molecular geneticist leading innovation in clinical genomics at the Broad Institute of MIT and Harvard. From driving gene curation efforts at ClinGen to advancing precision medicine, Marina's work is shaping the future of healthcare worldwide.
Hello everyone, I think we should start. Welcome to the PAPERI Bright Minds Series, where we learn and celebrate from exceptional individuals driving advancements in genetics, genomics, healthcare, and in Africa and beyond Africa globally. My name is Dr. Annette David, and I'll be the moderator for this session. To introduce the speaker, today we are thrilled to welcome Dr. Marina DiStefano, the Director of Clinical Genomics Operations at the Broad Institute of MIT and Harvard. She is a board-certified clinical molecular geneticist and Director of Clinical Genomic Operations at Broad Clinical Labs. She also directs the Broad Institute-based ClinGen Biocuration team and sits as a framework expert on 15 ClinGen expert panels across a range of different disease areas. Her research interests involve testing and setting curation standards in gene curation, variant curation, and disease ontologies. In this session, we'll explore Dr. Marina's journey, their contribution to clinical genomics, and the lessons they have learned along the way. We'll also open the floor to the audience for questions towards the end of this session. Dr. Marina, you're welcome. First, we'd like to know a bit more about your career journey. Once again, congratulations on your recent promotion. Can you share with us a bit about your journey in clinical molecular genetics and how it led you to your current role at Broad Institute?
Thank you for having me. I think that my desire to go into clinical genetics happened during my doctoral program. I was studying obesity, and the question in our lab in my program in biomedical sciences was, what is the difference between healthy individuals, so metabolically healthy individuals with high body mass index, and unhealthy individuals, so those individuals who are diabetic. So I was kind of adjacent to genetics at that point, but I realized, and was doing mouse genetics, but I realized that I wanted to be closer to the clinic. And so when I was looking for postdoctoral fellowships, when I finished my PhD, I started to apply to American Board of Medical Genetics and Genomics programs. And I found myself in a postdoctoral fellowship and variant scientist role at the Laboratory for Molecular Medicine, which is the lab at Mass General Brigham that Dr. Heidi Ream had previously directed. And so for a few years, I had learned how to classify variants, learned how to work on clinical cases, and then I transitioned into the ABMGG program at Harvard Medical School. And that was a wonderful program. I extended that over 3 years so that I could continue my postdoc with Heidi and also make it through the program. And so that program involved intensive genetics courses, shadowing in clinic, which was really inspiring and amazing. And then also working through a range of different clinical cases in the molecular laboratory. And then after that, you sit for a board exam. And when you're board certified, now I sign out genomes on a regular basis at the Broad Institute. And so that was kind of the journey that led me to where I am today.
Thank you. That sounds very interesting. And looks like you have worked in a variety of settings, from clinical testing labs at Harvard Medical School to the Jane Singer MyCode program. And can you share what have been some of the most pivotal experiences that shaped your career so far in clinical genomics?
Yes, I think I was very lucky to get involved with ClinGen as paid staff pretty much almost at the beginning of its creation. So ClinGen is the clinical genome resource. I'll talk a bit more about that, I think, further in the series. But it started in 2013, and I started my postdoc with Heidi, who is one of the ClinGen principal investigators, in 2016. And so I was able to get in at the ground floor, essentially, of when ClinGen was starting and really help with standard operating procedures, help with developing standards there, really start the expert panels while the expert panels were starting. I helped to start some of the first expert panels that we had, and now we have hundreds of expert panels. So I think that was really formative for me, and truly understanding when standards were still being formed, how important that they were, and really getting to network as a young trainee with international experts. I think that was extremely formative to me. And Heidi has always been a really supportive mentor. So I think that was the most pivotal thing. I met some really amazing colleagues, and the Geisinger team actually were ClinGen colleagues. And so I was able to transition easily to a faculty position at Geisinger after having worked extensively with them in ClinGen. So I would say that's probably one of the most formative experiences that I've had in my early training that's led me to where I am now.
Again, that's interesting. What I hear is you met the right people who were pivotal in shaping your passion for
Yes.
clinical genomics and mentors.
Yes.
Okay. We'd like to know a bit more about clinical genomics and gene curation. And in your role as a director of clinical genomics and operations, you oversee a team working on gene curation and exome genome analysis. Can you explain the importance of gene curation in clinical genomics and how it impacts patient care?
Yes, it's a great question. I think that gene curation is often underappreciated, but it's critical. Most people focus on variant curation, kind of forgetting in the first place that you need to establish whether a gene is actually connected to disease to be able to report variants in that gene, especially in an exome or a genome setting. And so I really do try to do a lot of mentoring and teaching and workshops to the importance of gene curation, and that it should always happen first. So when you're doing gene curation, you're asking if a gene is related to disease. There actually have been policy statements that have come out now by the American College of Medical Genetics that if you don't have a certain strength of a gene-disease relationship, you should not be reporting variants in that gene in a clinical setting. You could do it in a research setting, but not in a clinical setting. And so ClinGen, I participated in developing a framework for gene curation, a semi-quantitative framework where we look in the literature and we score cases from the literature to get to a certain strength level. So the ClinGen strengths, if people aren't familiar with them, are limited, moderate, strong, and definitive. And so the policy statement says that if you don't have a gene disease relationship that's moderate or higher, you should not be reporting that on a clinical test. And in a screening setting where you have healthy individuals who are looking for risk, you should really go even higher than that. And it should be a gene that has strong evidence or definitive evidence because people are going to take medical action related to your results. And I think in the past, it was more bigger is better. And so a lot of testing labs would put as many genes on their panels as possible, thinking that that's a better deal for clinicians. But then clinicians would get back a lot of variants of uncertain significance because there wasn't a good gene disease relationship for the genes. So I think that people have really started to focus on that a bit more. And for exome and genome analysis, it's really critical because you're looking at all of the genes in theory. And so you have to perform this gene curation essentially on the fly instead of the panels that have, a certain number of genes that are very well characterized. So gene curation is really important. I guess the takeaway of that is if you don't have a moderate or higher gene disease relationship, you have to have a GUS and a VUS, which is a variant of uncertain significance and a gene of uncertain significance. And if you have a moderate gene, the highest you can go in reporting variants in that gene is likely pathogenic. You can't get to pathogenic for variants in that gene. So gene curation is really important to do ahead of time so that you make sure that you have a link between the gene and the disease before reporting variants back to a patient.
So what I hear is gene curation is the foundation for variant curation. Uh-huh. I've never thought of it like that because many people nowadays focus more on variant curation. If you had these
Yes.
GWAS studies or case control studies where people try to screen for variants, people focus on finding variants of significance without focusing on the gene models. Oh, okay.
Yes.
And with your extensive experience in curation of genes and variants, how do you foresee the field evolving over the next few years, particularly in the realm of precision medicine?
It's a great question. It's a very exciting time, I feel, and there are many things for everyone to be excited about, but some of the ones that come to mind for me that I think we'll really be focusing on in the next few years are three things. And I would say the first one that I think of is MAVES, so multiplex um, analysis of variant effect. So we all know that we have a ton of variants of uncertain significance, VUSs. And if we could just have some functional data on these VUSs, it could really bump them one way or another. Like, are they benign? Are they pathogenic? And it can be expensive to make an animal model for every single one of them or do a cell culture experiment for every single one of them. So, really talented folks are going through generating high throughput screens in different genes and disease areas to really get an output of the significance or functional significance for variants. And so those are MAVES. And I would say certain disease areas are probably further ahead than other disease areas. For example, cancer, I think, has some really good MAVES. Some of the disease areas that I started in, they're starting to explore that now. So I started a lot of my training in genetic hearing loss. One of our collaborators in the expert panel is working on some MAVES there. And ClinGen is actually starting a group to evaluate, provide general recommendations for evaluating MAVES in combination with the Alliance of Variant Effects, the creators of MAVEDB, so the database that holds a lot of the MAVE data, and the IGVF consortium, so the Impact of Genome Variation on Function consortium. So I'm hoping that MAVES will be the future so we can really get some clarity on the function of certain variants. So that's the first thing. Another thing that I think is going to be really critical in the future, and ClinGen's already starting to explore this for our next round of writing grants, is machine learning and artificial intelligence and how that can help us in curation. Um, of course, there are a range of different ways that curation could be more efficient. We know that variant scientists and geneticists are needed because AI can't do everything, but especially as we do more exome and genome sequencing, there's just so much data to go through. We really need to find a way to be a bit more streamlined and efficient in our analysis. Um, and so I think in the future, we're really going to be exploring, at least within ClinGen, in a working group that we just started up to look at AI and machine learning. How can we use AI to efficiently find literature for us in a better way? We know that literature isn't extremely structured. Variants can be named 4 or 5 different ways in the literature. So how can we use artificial intelligence really to find that for us? And how can we use it to kind of summarize some of that evidence for us? So we review everything and then we write a nice summary at the end talking about a gene disease relationship. Can we use AI to generate that at least to get us started? And of course we would check it because, there are the caveats of AI that it might hallucinate and make things up. So we're really hoping to become a bit more efficient with those tools. So that's the second thing. And then I think another really critical thing for the future is personalized treatment. And I think that antisense oligonucleotides, ASOs, really are um, starting to become more and more usable and potential treatment, personalized treatment of the future. And so we are partnering with some organizations that are evaluating whether variants or gene disease relationships are amenable to ASO treatment. And we'll be displaying some of those curation results on our website in ClinGen so that patients and their families can look and see, like, is their particular disease area amenable to this antisense oligonucleotide treatment, and work with this group to maybe head towards a more personalized treatment. So I think high throughput assays, more efficient curation, and really personalized treatments through ASO are the 3 things that I think will be critical in our future.
And these are more for research applications, or they can be applied into medical applications, direct medical applications by patients?
So for antisense oligonucleotides, there are groups that have already treated patients with those antisense oligonucleotides. So I'm hoping that these will be clinically useful, MAVES, AI, and the ASOs, hopefully in the near future.
Yeah, exciting times.
Yes.
Yeah, we also would like to talk a bit about collaboration and leadership. And as a team leader at ClinGen, brought by Curation Core, how do you foster collaboration with your team And what are the key strategies that have helped you to manage such an influential group of experts?
Yeah, so the ClinGen team is highly collaborative, I would say. And so team fit is really critical for us. So when we're hiring, that's something that we ask about right up front. I tell everyone who interviews for me how collaborative our team is. So I manage a team of about 12 individuals at this point that work across all of the expert panels. And ClinGen is a bit unique in that it's highly volunteer based. So we have 2,000 plus individuals right now that a large percentage of those individuals are volunteers. So experts, curators, trainees, all volunteering their time to kind of forward ClinGen's mission. But we do have a small but mighty staff, and these staff really do become experts in all of our frameworks so that they can sit across all of our disease groups. And you have to work with so many different personalities, so many different people that you really do have to be a highly collaborative individual. And so when we interview, I ask about collaboration experience and how they work in groups together. And then I also will have chunks of my team interview individuals too to kind of assess fit with the current team to make sure that we're all on the same page and we're all working together. And I even though I have expert panels where maybe one staff member will take a little bit of the lead in that expert panel, they never work alone. I always have 2 to 3 staff members on every single expert panel so that there's a really good support system for them, and so that everyone can kind of share the leadership and share the responsibilities. So we try to be really collaborative in that way. And when possible, when I start groups, I try to really establish strong collaboration there, really open lines of communication. I think communication is really critical that and anyone who asks questions should always ask questions. There are no stupid questions. There are no silly questions. We're all learning together. And if it's a question you have, we probably have it too. So we've just really tried to have a lot of communication to make things so collaborative too. And then once I start an expert panel, oftentimes I'll try to step away or just step into the background and just provide oversight sometimes and really let my staff shine and learn along with the experts so that everyone is in a really helpful learning experience. And just encouraging active discussion and sharing of ideas. Because didactic sessions are great when you get on and you teach people, but that's not going to move progress forward. I feel like active discussions are really the most critical thing when you collaborate. So that's really how we try to make our team work, is just have a lot of communication, foster questions, and just active discussion.
Thank you. And as a follow-up, are there any collaborative tools that you recommend? Well, first, is your entire team work in person, or some of the team work remote from different places? And are there tools that you can recommend for teams, research teams, to foster collaboration? Yeah.
Yeah, it's a great question, and it's an interesting scenario. So I actually live in Pennsylvania, and my team all live in Massachusetts. So I actually manage them remotely from a distance, and I think that our collaboration is still great. I would say I've maybe potentially put a few more touch points in our lives than I would if I were physically in person, that they could stop into my office. So, for example, in the last couple years, I implemented an office hour. So we use Slack for a lot of our immediate communication. So like a messaging system, I would say, is pretty critical, especially if you're all remote. They're hybrid. They often will go into the office 1 or 2 days a week, and then we'll stay home the rest of the days of the week. So we have a couple things for that. We have Slack, which is direct messaging, which I think is really helpful. A bunch of different Slack channels. So I have one with all of the curators, and then the curators have one just as staff, like for quick curation questions. And then on Slack, actually, I have a little poll that automatically runs every week so that people can declare what days they'll be in the office. And this isn't to keep anyone accountable. Of course, it would be extremely hypocritical of me to say you have to be physically in the office this many days a week when I can't be in the office this many days a week. But it provides transparency so that if someone is thinking about coming to the office that week, they can look and see what day will the majority of other individuals be in the office? That way they can actually come and have some social interaction. So that's been really helpful that people can declare when they'll be in the office and spend time with each other. And that was based on feedback from them that they would like to try to coordinate as best as possible, but didn't know how to do it. So that was one piece. Another piece is the office hours, like I was saying. And so I felt that sometimes people felt that there was a bit of a high barrier to reach me for questions. And so they would really hold on to their questions until they felt that they were important enough. The questions, I mean, not the person. The questions were important enough to ask me, or that they were going to see me during our weekly check-in on Fridays. And there's no need to hold questions like that. So I really do try to encourage them to Slack me when they have questions, or we could have brief meetings. But then I also put on a weekly office hour where I just sit on a video call essentially, and anyone can enter and exit whenever they need to ask me questions. Slack has a really nice huddle feature that in our little Slack channel, I just hit the huddle button and it shows my picture that I'm sitting there. And then whenever anyone enters my office in that way, it shows their picture too. So if someone wants to enter at the same time as someone else, they would see the picture, or if not, they would wait until that person leaves and then they would enter. So that's been really helpful. I think a lot of the people who were afraid to bother me with questions have come to the office hour because it's already there. And if not, I just use it as focus time because sometimes they don't have questions or they have one-on-ones with me. So that's been really helpful. I also have monthly one-on-ones with them where I make sure and check in that their workload is still manageable and do they have any particular questions. So I think that's been really helpful. The ClinGen team is also really unique in that we are 3 separate grants at 3 institutions throughout the United States. And so we work very closely with individuals on the West Coast. So at Stanford University in California, we work really closely with individuals in North Carolina, so south of us. And we work really closely with individuals at Baylor in Texas. And so already we're extremely remote in that way because we're throughout the United States. And then on top of that, for our expert panels, we really try to get them to be international. And so those are all throughout the world. You're meeting people throughout the world. And so we really rely on video calls for those, I would say Zoom. And those have pluses and minuses. Every so often you get to meet the group of experts when you are at a meeting or an international meeting, and that's been really amazing. But on top of that, what can be helpful is when COVID happened, we could really continue our work because we were already entirely remote, at least with our experts. So I think a lot of the technology that has helped us has been like instant messaging systems and video calls. We also use a lot of shared documents. So we use the Google system at Broad, at least, where we'll have documents that we can all edit together in real time and share together in real time. And that's helped a lot too. So technology has been really great. I don't think this, would be possible 10, 15 plus years ago to have a team like this, but we've definitely made it work.
Yeah, and you have managed to create quite an impact. I think it's a lesson for most of us here who are trying to grow our careers and our teams as well. Yeah, thank you. You have contributed to at least 15 expert panels at ClinGen. Would you tell us about the role these panels play in advancing global understanding of human genetic variations and how they impact clinical practice?
Sure. Yeah, I'd love to give a little bit of an overview about ClinGen expert panels. So ClinGen, as I was alluding to, is a clinical genome resource. It's funded by the NIH, National Institute of Health. And we've existed now for 11 years at this point, started in 2013. We have over 2,000 individuals from almost 100 countries, I think, at this point, and many individuals are volunteers. And we have started up expert panels in different areas of curation. So I would say that the 2 areas of curation I focus on are the ones we've talked about. So variant curation and gene curation. And there are expert panels in both of those areas. So we have GSEPs, gene curation expert panels, and VSEPs, variant curation expert panels. And at this point in time, we have 16 clinical domains. And so examples of clinical domains are hereditary cancer, kidney disease, rhasopathies, cardiovascular disease, hearing loss, inborn errors of metabolism. So many of the main disease areas are covered at this point. And we have 57 gene curation expert panels as of this point across all of the different funded grants. And for gene curation expert panels, we really try to establish an international group of experts and with varied expertise. So we have clinicians, for example, we have laboratory scientists such as myself, geneticists, genetic counselors, people who are seeing patients, trainees, because they really help us with a lot of this active curation. And researchers who are doing functional data oftentimes because we know how critical functional data is. And for gene curation expert panels, we create a gene list, usually based on looking at what is offered in clinical testing panels right now in the world. And then we start to use ClinGen's framework that I talked about earlier to classify these gene disease relationships anywhere from limited to definitive. All of that gets done on monthly meetings. And once those curations are finalized, oftentimes my staff will perform those curations because they're experts in the framework. And then the experts in the disease area will provide them feedback. And then we'll finalize the curation. It's really critical for ClinGen to share curations as quickly as possible. So nothing is ever held for a paper publication. Pretty much when we finish a gene curation, it goes on our website so that people can use it. With all of the evidence that we scored, everything has to be publicly available. And the rationale as to why we scored it. And so those are gene curation expert panels. And then for variant curation expert panels, it's the same thing with an international group of experts with varied expertise. It's maybe a little bit more critical that we have individuals that work in laboratories because we need people who are very experienced with curating variants. So there's a bit of a requirement there. And then we take the general guidelines that were published in 2015 for variant curation, and we specify them in a disease area. And what I mean by specifying is there are general population cutoffs, for example, of like 5% for a benign variant and 1% in the population for a likely benign variant. That's usually very high for certain disease areas. So the experts will get together and say, how common is our disease? What frequency do we feel okay seeing a variant in the population and saying that's not disease causing? What are the specific phenotypes that we have to see in a patient with a variant to think that that's causative for the patient? What are the functional assays that exist in the field for our disease area and we would believe if we saw a variant tested in that way? So those are the kind of specifications that I'm talking about. So we work through all of those. That gets presented back to ClinGen, to a working group that reviews them. We pilot all of those specifications with like a truth set of variants that we know, like these ones are pathogenic, these ones are benign, and are our rules working appropriately for those variants? And then they all get published to ClinVar, so the variant database from the National Center for Biotechnology and Information, as expert panel assertions, so the highest level of assertions, which is 3 stars if people use ClinVar. And then they're recognized by the FDA in the US. And that's also why our paperwork is a little bit heavier, because we are FDA recognized. And so those are the 2 sets of expert panels that I work with, and there are currently 71 variant curation expert panels, so in a range of different disease areas. And I will put a shameless plug out that we're always looking for new members for our expert panels. So if you go to clinicalgenome.org, you can take a look at the expert panels that we have. And we have a range of different volunteer activities because volunteers are so critical. So we have a new survey to collect experts. Based on the workshops, the international workshops I've done, that there are plenty of experts that we've never met, and we'd love to meet new experts. So you can fill out your expertise in that survey, and we pair you with an expert panel. Or if you're more junior and would like to learn about curation and really network, we also have a volunteer opportunity where you can say that I want to curate in an expert panel. And then we take you through all the training that you need. I lead a lot of those trainings. And you can choose what type of curation that you want to do and maybe what disease areas you're interested in. And then you'll get paired with expert panels as volunteers. And we've paired hundreds of volunteers now. So definitely just a plug if you're interested to volunteer.
Thanks. Can you repeat the website? I can type it quickly in the chat.
I can type it in the chat myself, I
Yes,
think.
please. So for anyone who is following, if they're interested, they can get it quickly. Thanks. I see it already.
Yeah, and let me find. So on the here's the one. I'll stick this in the chat too. This is if you're an expert.
Yes.
And then if you are just interested in volunteering and you're more junior, this is the other one.
Okay. Thank you. We also would like to talk a bit more about the challenges and opportunities in clinical genomics. And first, we'd like to know what are some of the biggest challenges you face in your work, particularly in the context of clinical genomics operations, and how do you go about solving them?
Yeah, it's a great question. And I think a lot of the challenges that I face are often the challenges that the clinical genetics community at large faces. And I think a really big issue that we have right now is lack of access to global data. We know that many of the publications have been done on European populations, and that's just not helpful. We've saturated all the information that we have on European populations. It's not giving us any more useful information. And at least in the clinical setting, I'm not just getting European patients. And so a lot of times when I have individuals who have a background that hasn't been published on, that is not represented in biobanks, for example, I really have a lot of difficulty interpreting their case because they just end up with a lot of variants of uncertain significance because we don't have any publications about it. We don't know if they're frequent or not. So I think that's been a really big challenge and a lack of equality there. And I think that we are getting better. We're trying to be as collaborative as possible and recognize that data needs to be connected, but not that one person should take all of the data and put it in one place. That doesn't make any sense. We really need to have federated data, which means that everyone can hold on to their data, but they can all search it together in the middle. They kind of call it like a wheel and spokes model. And so I'm lucky in that I get to work on a daily basis almost with the Nomad team, which is one of the really large population databases in the world. And they're working very hard to get a federated Nomad, which means that everyone can hold their data in their countries, but can search it so that we all can see all of the wonderful biobanks that people have generated throughout the world, because people do have some really wonderful biobanks. So I think that is one active challenge that we're very aware of, and we're all trying to work together to reconcile this challenge, but I do still think we have a little bit of a ways to go to get there.
Yeah, I think one of the biggest challenges in advancing genomics in Africa is that lack of data. And many populations remain unsequenced, their variants are not classified. And I'm not sure if there is specific advice or source of resources or support for research groups which are trying to generate more human genomic variation data from Africa, for example, and other less represented countries. Is there any kind of resources or advice, or are there initiatives which are trying to increase the representation of genomic data, human genomic data?
Yeah, it's a great question. So I know ClinGen has actively partnered with H3 Africa before. And I would say the biggest connection I have had in trying to bring resources to other countries is education. We really have been trying to focus on international workshops, kind of like a train the trainers model that I would love. I have workshops, and I will train a group of individuals extensively who are maybe like some of the leaders in this field, or some of the first trainees, to really have how to set up a robust curation infrastructure. And that's maybe like my particular expertise in that area, is to make sure that you have like a really strong basis for curating. But I know that there are some really good collaborations both in industry in the US, like with reagents making reagents more available and more reasonably priced. And genetic counseling programs. I know there's really a lack of genetic counselors, I think, throughout a lot of Africa. If you have a small but mighty team, is my understanding, like um, less than 20 individuals or something, and they're all amazing. So I think my biggest connection is with education, but I know that there are some efforts with other individuals trying to really bring resources to your teams so that you can do the sequencing and the reporting and generate more data. I think another thing that's really critical that you're probably aware of is building trust with the community, particularly in populations that have been vulnerable and abused. In the past, I think it's really critical to have those community representatives. And so we have some really good examples of that now in the US. And so I think that's also an additional piece of education and working actively with your communities to build that trust, and also keeping your data in your country, like I said, to have like that federated model, because I think that builds the trust too. There's a really big fear of people's data being shared or stolen or leaked, and it's a legitimate concern. So I think there are many pieces that lead to increased access to testing, and the piece that I try to really focus on, because it's in my expertise, is education and training.
Yeah, thanks. And I really agree with you in terms of building trust with the public, because also people now are concerned with when profit is generated from the data, because some companies can benefit by developing diagnostic tools or therapy tools, and then they're commercialized, and the
Mhm.
data where people give the data, but they do not benefit from the data at all. So I think
Yeah.
that's one of the areas also to really try to straight out to the public before collecting the data and collecting samples and sequencing.
Yeah.
Yeah. But also, what are the exciting opportunities that you see on the horizon for clinical genomics, particularly in terms of global collaboration and advancing healthcare through genetic insights?
Yeah, I think the thing that I'm most excited about is the focus on global engagement in ClinGen now. As I mentioned, I've been really lucky to get to lead curation workshops around the world. I feel like when we first started ClinGen, we were very focused on gathering as many experts in the United States, just because we needed to become a bit stronger and a bit more stable. And now, as we've hit 10 or 11 years of existence, we really are trying to outreach. We've stopped attending a lot of US conferences just to save our travel budget to be able to go internationally and do these workshops and recruit experts across the world that we might not have had access to before. So I've been really lucky in the last couple of years to perform workshops in Latin America, and then also workshops in the Middle East, based on just the collaborations of meeting people along the way. And so I'm really excited about that future global engagement. And I think both myself and my colleagues in the Broad Geisinger grant and ClinGen will continue to do workshops internationally with folks. I know that my colleagues are going to South Africa to the Hugo meeting in a couple weeks, actually, and we'll be performing a workshop there. So we're just really excited about all of this global engagement. I think it's really thrilling to get to meet people in person. I know that a lot of times when I have events like this, or much larger training events, for example, that it can be a really big barrier to meet someone and ask questions and really make that face-to-face connection. And I just think those barriers are taken away when you go in person. I met such wonderful folks in Latin America and Argentina and Brazil when I went, and Qatar also when I went. And I would love to keep doing that, because it really excites me to meet people across the world and um, find out what we have in common and find out what we can learn from each other. It's just been a really great experience.
Yeah, technology does help, especially with like now we are able to have this webinar via technology. It would have been impossible in the past, but I do agree that sometimes that face-to-face connection is important. Is there a place on your website or somewhere? Is there a place where people can see? Do you have a schedule maybe for these international workshops that people can anticipate? Do you have a
It's
schedule
a good
posted somewhere? Yeah.
it's a good question. We have a lot of schedules that we advertise on our social media accounts, I would say. So we're very active on LinkedIn And I think we've maybe started a Blue Sky account now, and maybe one or two other things. And so we advertise on those. But the other nice thing is, let me find it. Oftentimes we'll share the resources that we did for those workshops. So for example, the workshop I did in Qatar in December, all of those PowerPoints and different training modules we made are publicly available on our website. Let me see if I can find them. So link in the chat. Let's see.
Yes, that would be great.
Oops. Yes, there is a section, events and conferences, that has a list of all of the events and conferences that we've had so far. So I can stick that in the chat.
Yes.
But we have a lot of educational resources from those that we have. And we also actually have a YouTube channel that we record a lot of things and we put on there for trainings. ClinGen resource, hold on, ClinGen
Yes. People can take them.
resource YouTube channel.
Okay. Thank you. People will check them out and see what the resources they can get. But also we'd like to talk a bit more about the impact on next generation of scientists. As a mentor and a leader, what advice would you give to young scientists who aspire to make a difference in the field of clinical genomics or molecular genetics?
Yeah, it's a great question. And I would say, and maybe this is just because I've been involved with ClinGen from the very beginning, that collaboration is so important. We can't all do this alone. We have realized that we can't just all keep all of our data in separate silos and not share the information that we have, because then that will result in unequal patient care. And so my suggestion is to try to collaborate as much as possible, volunteer for things, reach out and become a part of ClinGen, meet some of these really senior individuals, and ask them questions. Try to learn as much as possible from everyone that you meet. Get involved and get involved early. One suggestion that had been given to me that I think has been really helpful is, if you have societies, for example, volunteer for positions and committees as early as you can, because if you get involved as a trainee, you really start to work and collaborate with a lot of those senior individuals that are leading that society. So for example, I started volunteering with the American Society of Human Genetics when I was a postdoc. And I went from the communications committee to the program committee, which, sets one of like the top programs in the United States for genetics meetings. And now I'm on the awards committee that chooses, some of those really big national awards. And I've met so many amazing people throughout that. And it can be a lot harder sometimes to get involved with that when you are a more senior individual or maybe a mid-career individual, because everyone's trying to get involved then. Whereas if you get involved when you're a trainee, a lot of times people are not thinking about getting involved then. And so you get that kind of early access to a lot of those senior individuals. Don't be afraid to ask questions. I think that people are often afraid that they're going to be seen as stupid questions or, not thoughtful questions, but asking questions really teaches you and helps you learn and helps you formulate questions, and also, exposes you to those individuals. And then I think maybe the last thing I like to say about that is, high quality work often speaks as loudly as loud words. There are so many people who like to just talk about what they might do, but you should just do things sometimes. And one example of that I have is pretty much right when I started as a postdoc. Everyone was talking about how they needed to make a standard operating procedure for gene curation. And, there was a lot of talk about it, and I volunteered to do it. And it was done within a couple weeks. And I think that held a lot of weight in the community in ClinGen, is when you have concrete results that show that you produce good work, oftentimes that will, give more trust than just sitting there and talking about how you really want to produce good work. So collaborate, produce good work instead of just talking about it, and ask questions and get involved in opportunities and societies as early as you can.
Excuse me, in this context, I would like to say something about Dr. Marina. No one knows this. Dr. Marina helped me many times in many webinars. Actually, we succeeded to present in PAPERI. So thank you so much. So take her advice seriously. Collaboration. She is the one who worked and still working with me to facilitate the webinar. Thank you, Dr. Marina, really.
Of course.
And that is very solid advice. I wouldn't add a thing. And as an early career myself, I think I've seen some points to strengthen, but some new things to start. I think we still have a bit time for one more question, and then we can read some questions in the chat, and otherwise we can continue. Still on the topic of advice to the younger generation, how can we increase the accessibility of genomic testing and genetic counseling to undeserved populations, particularly in Africa?
Yeah, it's a great question. And I kind of have talked about that a little bit. I think there are a couple different aspects. Like I said, my aspect that I can focus on and provide the most advice is education. I think it's really critical for people who are trying to educate other people, so like myself, to go physically to those places, have workshops, train as many individuals as possible who are willing and able to train future generations. So I think those train the trainer workshops are so critical, really to give experience on open source materials and open source platforms. For example, there are so many platforms now. So like ClinGen's platform is entirely open source to classify variants. So really to just be able to share that knowledge and educate individuals. We also have career series. And I know that when we've done workshops, for example, in Africa in the past, we've talked about genetic counseling and the experiences that genetic counselors have had and the types of training programs that we have in genetic counseling. So I would say really the ability to train the trainers instead of just coming back over and over and over again to train people, I think is really critical to establish that first generation who comes into the ground floor, kind of like I did in ClinGen, and can really become formative in your genetics experience in different countries, I think is really, really critical. And, of course, keep those lines of communication open. I've met such wonderful colleagues in Argentina, for example, that I talk to all the time. I see them at other meetings. When they have questions, they email me and we have a really great discussion or hop on a call, things like that. So I think that's the thing that I can offer the most is education sessions. But of course, recognizing that reagents need to become more available, reagents need to become less expensive, and sequencing technologies need to be able to be less expensive and more accessible to everyone in every country. And you can really only go so far without this type of funding and financial support. And so recognizing that those are really critical too, but that my critical piece, I think, is bringing the training and the education to as many people as possible.
Absolutely. We still have some time, and maybe we can go through the questions we have in the chat, and then if we have more time, we can finish with a few questions. And
Sure.
I'm going to give myself some chance. So for the first question to come from me. In Tanzania, we co-founded Tanzania Human Genetics Organization, and one of the things we are working on is rare diseases. We read other countries have found these gene associations or variant association to rare diseases, and I was wondering whether your group has any work they have done on rare diseases.
Yes, I would say actually most of my work is in rare diseases and most of ClinGen's work is in rare diseases. We are trying to do some new work a little bit in common diseases, but I think pretty much all of our frameworks and all of our expert panels are now focusing on rare disease. If we're talking about really rare disease, we also have some expert panels for that. So one of my favorite expert panels, don't tell the others, is the syndromic disorders expert panel. And so this expert panel is one of the expert panels that curates the diseases that are really across all body systems. Because, you know, I told you in the clinical domains, we have like cardiovascular, we have skeletal, we have kidney disease. So if there are some really rare syndromes that have features across almost every clinical domain because they're very severe, we actually take them to that syndromic disorders expert panel. And it's been a wonderful learning experience. We have a really vast array of different individuals, dysmorphologists, clinicians, researchers who try to become experts as much as possible in as many rare diseases as possible for this. But I learn about a new syndrome every time I'm on that call because they tackle new genes and new syndromes every single time. But I would say almost everything in ClinGen is rare disease at this point.
That's very interesting. I'll be sure to check the website for more resources. Thank you.
Yes.
Now in the chat, the first question, I think you have addressed it, but I'm going to read it anyway. And if you have anything to add, you can add. For scientists and clinicians who are interested in contributing, what does the process of becoming a ClinGen volunteer look like? What skills or expertise are most needed, and how can volunteers make a meaningful impact?
Yeah, it's a great question. So I would say we have two volunteer pathways at this point. So the one, which is the newest, is if you're an expert. And when we say expert, we have some definitions on the website if you're unsure. But I found that in my travels, there were people who were performing all the clinical genetic testing for a country, and they did not feel that they were experts. You are an expert. If you have extensive functional experience in functional assays, if you've been researching a disease area your entire career, if you are a genetic counselor who's seen individuals in clinic, you are an expert in that disease area. And so there is an application process for experts. And for that process, you include kind of a bio sketch or a CV or a resume, and then also answer a series of questions about your expertise so that we can most correctly pair you with with expert panels. And then you also will mention what expert panels you're interested in joining on the ClinGen website, and they're all listed there. And for experts, most of the time that involves about an hour or two of commitment a month. We have expert panel calls once to twice a month. They're 1 hour long. And this really is to watch some of the curators perform the curations, either my staff curators or the volunteer curators, and provide your expertise in the disease area. So they'll essentially teach you the framework, but then you will teach them about the disease area and weigh in along with the discussion of other experts. So that's the expert volunteer process. And for curation, oftentimes these are individuals who really are not, later in their career. Maybe they're trainees, maybe they're early career individuals, people who would like to learn more in a disease area and who might have a bit more time to participate. They can volunteer to be curators in the expert panels, and you can choose what type of curation you'd like to do. I only talked about two types of curation, so gene curation and variant curation. We have other types of curation too. So for example, dosage curation is kind of like a cross between gene and variant curation that is evaluating copy number variants, essentially. So if you remove an entire section of a chromosome, for example, is that going to cause disease, haploinsufficiency? Or if you add an entire chunk, triplosensitivity, is that going to cause disease? So they also accept volunteer curators. We have the actionability curation group that evaluates once a gene is associated with disease, how actionable are the conditions if you were to screen for them in a healthy population. And they also accept curators. So you can choose when you fill out the survey what type of curation that you're interested in. And some curation types require potentially a bit more experience than others. I would say in general, the preference for variant curation is that you've done a little bit of genetics and or variant curation before. So I would say gene curation is a really good beginning curation volunteer group to go with. And the commitment is often 6 to 8 hours at the most a month. So it is more than an expert. But oftentimes you will be asked to prepare a curation for one of the meetings, maybe like once every couple months, because most groups have enough volunteers. But once you fill out the volunteer survey, there is a list of training materials that you review, and then we also will have virtual training sessions. I lead many of the virtual training sessions where we show how to use our web interfaces to curate. So kind of two different ways to volunteer in that way.
Thank you very much. Those are so many resources and pathways for people to learn. And that is all the time we have for today. Maybe if you have any closing remarks, any last words, closing remarks before we end the webinar.
Just that I've really enjoyed talking to you. I think some of you do have my contact information, so if anyone has any further questions, feel free to send them along to me. And I am certain that we will meet in other avenues. The world is a smaller and smaller place every day, and I just really enjoyed to have this chat with you.
Thank you very much for sharing your incredible insights and experiences with us. And your work is truly inspiring, and we are honored to have you today. And to the audience, thank you everyone for attending this session, the Bright Mind series. Please stay tuned for more sessions featuring exceptional leaders and innovators who are shaping the future of science, healthcare, and education. Please check out PAPERI on social media, especially on LinkedIn, and follow us and ask questions and interact. Thank you very much, and have a good day, everyone.
Bye.
Thank you, Dr. Marina.
Bye-bye.
Bye-bye. Thank you, Dr.
Great.
Anis.
Great
Bye.
job.
Bye-bye.